Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's biology lesson, continuing our unit on evolution. In our last lesson, we learned about a few more theories of evolution, including the eternity of life theory, cosmozoan theory, and biochemical theory. In today's lesson, we will learn about the experiments conducted by Alexander Oparin and Stanley Miller to explain more about the biochemical theory. In the 1920s, Alexander Oparin and John Haldane were working on explaining the theory of biochemical evolution. They had three conclusions. One was that the early atmosphere of the Earth did not have oxygen like it has today. The second conclusion is there was a large supply of energy from lightning and UV light. And lastly, the lack of oxygen and supply of energy would provide the energy for reactions that would produce many different molecules, such as amino acids and sugars. The amino acids and sugars were found to be the molecules needed for the development of life. These molecules could be formed through a reaction between the gases hydrogen, carbon dioxide, ammonia, methane, and water vapor, and an energy source. After some observation and experiments, the scientists concluded that the molecules needed for the development of life could have been made without the existence of life under certain conditions. Oparin and Haldane hypothesized that UV light, along with lightning, provided the energy that would produce the first simple building blocks necessary for life. This is called the biochemical theory. Oparin's original idea was to observe and explain how complex molecules arose from simple chemical reactions. He proposed that the molecules could have gone through a series of reactions, making complex molecules. The molecules made from the biochemical reactions could have formed coacervates or precells. The coacervates were able to absorb amino acids and sugars, also called organic compounds, from the environment. A plant uses the environment around it to breathe and make food. This is known as photosynthesis. The coacervates went through a process that was similar to this. According to Oparin, the coacervates may have eventually evolved into the first simple cells. Pre-cells would then be the ancestors to the cells we know today. Students, let us review. For our first activity, please answer the following question. What was Alexander Oparin's theory trying to explain?
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. Welcome back, teacher. If you answered that Oparin was trying to explain how complex molecules arose from simple molecules, then you are correct. Well done. Let us move to the subject of Stanley Miller's experiment. So far, we have discussed the theories that Oparin and Haldane suggested about the biochemical reaction that occurred to form life on Earth. However, there was yet to be any proof of this theory. In 1953, Stanley Miller conducted an experiment that confirmed Oparin and Haldane's theory. Miller's experiment simulated the Earth's early conditions in a closed-off structure. He sent electrical sparks through the structure that contained the gases hydrogen, methane, carbon dioxide, ammonia, and water vapor. These are the gases that were present in the Earth's early atmosphere. The reaction between the gases produced complex molecules. And as we know, these molecules are the required building blocks of life. Miller found that if he left the experiment untouched, the simple molecules would turn into more complex molecules, such as amino acids, which were needed to form proteins, pentose sugar, which were needed to form nucleic acids, hexose sugars, which were needed for respiration and to form starch and cellulose, and hydrogen cyanide. Since Miller's experiment, there have been other findings made about biochemical theory. In 2008, scientists tried to duplicate Miller's experiment using new findings and technologies to achieve detailed results. They simulated reactions like volcanic eruptions in the experiment. The new reactions produced even more amino acids and other molecules that helped the scientists understand biochemical theory better. Students, let us review what we have learned about Stanley Miller so far. What gases did Stanley Miller use when he simulated Earth's early conditions? I will give you a list of 3. Please choose the 2 that are correct.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. Welcome back, teacher. Did you choose the 2 correctly? If you said that Stanley Miller used the gases ammonia and methane in his experiment, then you answered correctly. Hydrogen cyanide was a product of the reaction. Well done. This concludes our lesson today. We learned about Alexander Oparin's theory on biochemical evolution, and we learned about Stanley Miller's experiment that proved this theory. Until next time, thank you, students. Thank you, teacher.